Good afternoon, dear students. This is the diagrammatic explanation of immunological technique or immunochemical method used for selection of clones. In the part 2 slide of selection of clones, we have discussed about colony hybridization technique and number 2 immunochemical assay. So this is the diagrammatic representation of immunochemical assay. The theoretical aspect we have discussed in the second part. Okay. Now you can see here you have a master plate. Okay, the master plate is having certain colonies on it, or the transformed cells are allowed to grow on a master plate. Then the colonies on the master plate are transferred on a nitrocellulose membrane. Okay, the nitrocellulose membrane is exposed to chloroform vapors. The nitrocellulose membrane is exposed to chloroform vapors. See, the colonies on master plate are replica plated on nitrocellulose membrane. They are allowed to grow, and then the colonies on the nitrocellulose membrane are exposed to chloroform vapors so that the bacterial cell gets lysed and proteins are extracted out. So you can see here bound proteins, okay, which are bound to the matrix. Then what you have to do is you have to incubate this nitrocellulose membrane with primary antibodies. Okay, so treat the matrix with primary antibodies. Now you can see here in this diagram the primary antibodies. Here you can see the primary antibodies combine with the antigenic protein produced by those cells which contain the recombinant DNA, which contain the foreign gene inside it. Then okay, now you have to again incubate this matrix with the secondary antibody. The secondary antibody may be enzyme labeled or it may be radio labeled. Here in this diagram we have shown that the secondary antibody is enzyme labeled. So you on the surface of this matrix, after washing it, after treatment with primary antibody, you have to wash it to remove the unbound antibody, then add secondary antibody. Now we can see here the secondary antibodies have combined with the primary antibodies. Now the secondary antibodies are enzyme labeled. That is why we add a substrate. The enzyme combines with the substrate, breaks the substrate, and colored product is formed. Where colored product is formed? At the place where antigen antibody reaction has taken place. That means antigenic protein is produced by the desired gene which has combined with the primary and secondary antibodies. So the place where you can see a colored product, that colony is selected from the master plate. Okay, so that colony you have to actually compare, the position of the colonies, and the colony is selected from the master plate and subcultured for future use as the colony formed by the cells which have recombinant DNA, foreign gene inserted inside it. Okay, now so you can see here colony expressing functional enzyme. Okay, so you have a colored product. You can see the formation of a colored product here. So this is how you select the cells that produce the antigenic protein or desired protein by immunochemical method. But if your secondary antibody is radio labeled, then what? Okay, so here you can see incubate the filter with primary antibody. Okay, wash and then incubate with radio labeled antibody. You can see here blue colored ones are primary antibodies, okay, which have combined with the antigenic protein produced by the recombinant cells, and then you add iodine labeled secondary antibody. Okay, secondary antibodies are I125 labeled. Okay, so the iodine labeled secondary antibodies are added. They combine with the primary antibodies which are bound to the antigen. So you get a fusion protein is formed. Fusion protein means antigen, primary antibody attached with secondary antibody. So you can see here. Right. Now because it is radio labeled, what we do is autoradiography of this membrane is done. What we do? Autoradiography of this membrane. And you can see a dark colored spot of the colony that has the recombinant DNA inside it. Now this X-ray image is compared with the master plate. What we do is the X-ray image is compared with that of the master plate. Right, and the colony from the master plate is selected and used for further use or for further production of the antigenic protein. So this is immunochemical assay. So this is the diagrammatic representation that we are discussing here. Okay, the theoretical part we have discussed in the earlier presentation. So that is all about selection of clones by immunochemical method.